Mhm. Okay, so we'll continue here. Um, cell structure. Okay, so yesterday we have covered some basics of microscopy. Okay, so comparing like light and electron microscope. Okay, so I want to do a recap for now. Okay, and then so we started looking at different cellular structure. Okay, so yesterday we have also looked at nucleus and ribosome. Okay, so and also mitochondria. Okay, so let's just continue with chloroplast. Okay, so as you are very familiar, okay, chloroplasts are only found in plants. Diameter is about 3 to 10 micrometer. Okay, one of the key features of chloroplast is that it has a double membrane. Okay, so it is seen here. Okay, so and also what you will realize from this diagram. Okay, so inside the inside the chromosome, inside the chloroplast. Okay, so there are these membrane sacs which are flattened. Okay, so each of them, okay, each of these is called thylakoid. Okay, so just a little this. Okay, so a stack of it is called a granum. So this whole stack. Okay, so this whole stack is a granum. Okay, so each of those this is this of a membrane sac is actually thylakoid. Okay, so of course again this kind of additional membranous structure is to increase surface area for photosynthesis. Okay, so the inner membrane is shown into thylakoids. Okay, but and inside the thylakoid is photosynthesis chlorophyll. Okay, so which is the key color pigment for photosynthesis. Okay, and outside here, so this liquid filling the space of the inner space of chlorophyll is stroma. So in mitochondria, it's matrix. Okay. So here it also contains enzyme for photosynthesis. Okay, in the stroma. And it also has its own circular DNA and 70S ribosome. Okay. As we learned yesterday, 70S ribosome is mainly found in prokaryote and in fact circular DNA as well. In high school, you may have come across the term plasmid. Okay, that's a circular DNA. Okay, if you remember, for SPM syllabus, you learn it for genetic engineering. Okay, so yeah, circular DNA is a plasmid. So again, these two are characteristic of what we call prokaryotes. Okay, so prokaryotes. So yesterday we learned about the term eukaryote. Eukaryotes are cells or basically organisms with true nucleus. Okay, so you must have come across this term as well. Okay, so by true nucleus, it means they have membrane bound nucleus. Okay, but these two are characteristics of prokaryotes, but somehow they're also present in eukaryotic cells, okay, like our cells. So, like I mentioned yesterday, okay, so there's theory suggesting that chloroplast and also mitochondria were originally two independently living prokaryotes, but somehow at some point during the evolution, they live symbiotically with the eukaryotic cells, okay, and then stay there forever.
Is that a theory or is that like a?
It's a theory, it's a theory, it's a theory, it's a theory, okay. So chloroplast is a cell photosynthesis, as you already know, okay. So next is ER, the endoplasmic reticulum. Okay, so this is ER. Okay, so ER is a case system of flattened sites. Okay, so you notice that case again, if you look at this image, okay, as mentioned yesterday, some of it can be an extension of your nuclear envelope. Okay, so it's an extensive network of membrane. Okay, so you can see that this membrane, they're interconnected. Okay, at some place. Okay, of course, this is just a section of it. Okay, so they are all interconnected. Okay, so for the parts which close to the nucleus, okay, you can see that they have ribosomes sitting on it. Okay, so because of the ribosome, the granular surface make them call lead to the name rough endoplasmic reticulum. Okay, and as you probably so it is a provide large surface area for protein synthesis. Okay, again, because of the large membrane folding, it provides larger surface area for protein synthesis. Okay, it provides a pathway for transfer of material, especially proteins, through the cells. Okay, so we are going to learn that. So it is actually through this membranous structure where protein is made, and then after that they will pinch off. Later I'm going to show a video for that. Okay, and then they form a transport vesicles that transport protein to Golgi apparatus. Okay, and smooth ER as opposed to rough ER. Okay, so simply because they lack ribosomes. Okay, and they play a slightly different role, which is make lipid and steroids. Okay, so the rough ER are for protein synthesis, and then smooth ER are for lipid and steroid synthesis. Okay, and unlike the rough ER, the smooth ER is sometimes it can be an extension from the rough ER. Okay, so they are not directly connected to the nuclear envelope typically. Okay, but that's the structure that you will see. And just FYI, rough ER is normally the protein that is synthesized typically for exports. Okay, so basically whatever proteins, most of the proteins synthesized at the rough ER are mostly for extracellular use. Okay, so basically you have to transport it outside. For example, if you secrete insulin or any hormone that's protein in nature, okay, so they are synthesized by the rough ER. For other proteins which are to be used within the cell, the ribosome, yesterday we also learned that your ribosomes can be freely floating in the cytoplasm. Okay, so ribosomes which are freely floating in the cytoplasm are responsible for synthesis of protein to be used within the cell typically. Okay, so next is Golgi body. Okay, or sometimes we call Golgi apparatus or Golgi complex. Okay, so just several terminologies. Okay, so unlike rough ER, okay, or the smooth ER, okay, so it's a stack of flattened membranous sacs. Okay, okay. Lysosome, which one? Oh, SOM, yes, SOM, yes. Lysosomes, okay. Okay, so let's have a look. So this is your Golgi. Okay, so so there are several features which distinguish your Golgi from the rough ER. Okay, so although they are both membranous structure, okay, so you notice that so it's a stack of membrane sacs, so they are not interconnected. Okay, I want you to know that they are not interconnected, and typically you will always see vesicles budding off from this Golgi. Okay, so you can see we say that the vesicle pinch off. Okay, so pinch off from the stack. Okay, So they move to the cell surface membrane, fuse the membrane, okay, and release your content again. So it is part of the transport system, like I mentioned just now. Okay, proteins are to be used for outside of the cells. Okay, they are actually synthesized at the rough ER. Later, as depicted in this diagram, okay, we do see that whatever proteins that is synthesized here, they will pinch off, okay, and then they will come to the Golgi for further modification, okay. Later, we look at what the modifications are, okay. So here, again, there are lots of enzymes doing the modification, or we say doing sorting and packaging. So here, by sorting and packaging, so they sorting by sorting it means, okay, so this, okay, for example, there are particular proteins which have similar function, or maybe the same proteins will be grouped together, for example, or all of these, because at one, your cell is a very, very busy factory synthesizing all kind of proteins at the same time. Okay, so you imagine that at the Golgi apparatus, there are different kind of proteins. Okay, so sometimes, okay, so let's say insulin will be all sorted together, packaged together, and then to be sent out of the cell. Okay. So, and then some of them will actually, okay, for example, if you, what is lysosome? Can someone tell me? Do you remember what's lysosome?
To digest something.
Okay, so lysosomes contain hydrolytic enzyme, okay, which helps to break things down, okay. So for example, if any bacteria or any foreign material that's ingested, or if you have any organelles which are old, okay, useless, okay, they will actually eventually be digested by enzymes contained within the lysosome, okay. So anyway, a lysosome is also formed from the Golgi, okay. And so here, so some of the function, okay, so modification of proteins by adding carbohydrates to form glycoproteins, okay. So there's also modification going on here. So as you probably learned in high school as well, your glycoproteins are, where are glycoproteins normally found? On the plasma membrane, okay. So, and okay, so they are normally found on the plasma membrane, and also some proteins which are to be exposed on the cell surface, okay, or to be embedded in the cell surface are also synthesized by your rough ER, okay. And then they produce secretory enzymes, okay. So anything to be secreted, for example, your digestive enzyme, okay. And in fact, and other than that, hormone as well, okay. So they may also go through your Golgi body, okay. So then they also function to convert sugar into cell wall components in plants, okay. So that's the case in plants, okay. When we come to the chapters on cell mitosis, we'll come across this, okay. And transport, modify, and store lipids, okay. So yeah, it is also involved in the modification and transport of lipids. Okay, so because you will also have lipids coming from your from the smooth ER. Okay, so in animal case, so Golgi vesicle may form lysosomes. So note that lysosome is a structure that is unique to animal but not plant cells. Okay, so you can see that in general, the whole Golgi is what we call a sorting. It's a modification and sorting and packaging center. Okay, so proteins and lipids are modified here, sorted, packaged, and then sent out of the cell. Okay, so next is lysosome. Okay, so lysosome, you probably know the word lys means break things down. Okay, you have come across the term electrolysis, hydrolysis. Okay, it all means break things down. Okay, so lysosomes is actually an organelle that is responsible for breaking down protein or breaking down things. Okay, so it's a single layer membrane. Okay, it's a single membrane organelle. Okay, without any internal structure. So diameter is about 0.51 to 0.5 micrometer. Again, under a typical light microscope, you wouldn't see it. Okay, because the membrane is very, very thin and the size is also very small. Okay, so you can't really see it under the light microscope. Okay, so they are formed from Golgi apparatus. Okay, so they're formed from Golgi apparatus and contain digestive or hydrolytic enzyme. Okay, so the main function is to break things down of unwanted or worn out organelles. Okay, so even organelles is too old or no longer or dysfunctional, they will be broken down so that you can disassemble it, and then whatever protein, amino acids or carbohydrates can be reused. Okay, and digest material ingested by phagocytic cells. Okay, such as white blood cells. So I believe you have learned the term phagocytosis. Okay, so phago or phage means eat. Okay, so we may come across the term macrophage. Anyone remember what's macrophage? You remember, have you seen macrophage before? Macrophage. Macrophage, have you learned the term macrophage before? So macrophage is a white blood cells which function to eat things up. Okay, so yeah, and it performs phagocytosis. Okay, so typically you would see it forms some membrane projections surrounding whatever, let's say this is a bacteria, and then engulf it, and then it will pinch off and then form a vesicle. So this vesicle containing whatever that's been eaten is called a phagosome. Okay, so this phagosome is ultimately, as you look at see in this diagram, okay, it will fuse with the lysosome. Okay, and then the hydrolytic enzyme inside the lysosome will break down whatever that's been eaten up by the cell. Okay, and it is also involved in a process called autolysis. Okay, so basically cell breakdown. Okay, so cells, our cells have a process called programmed cell death. Okay, when a cell is old, okay, they know that okay, it's time to commit suicide. Okay, so and part of that is contributed by the action of hydrolytic enzyme, which just break everything down. Okay, and yeah, so actually like cancer cells, they actually turn off their suicide program. Okay, so that's why they don't die. Okay, so and also in a head of sperm, okay, in high school you probably have learned that in your sperm or the head also you have a very large nucleus, and then there is a membrane sac containing lysosome, so or called acrosome. So it is involved. Okay, so it is required for breaking the dense layer of matrix around the ovum. Okay, so yeah, okay, for design, okay, digesting path to the ovum. Okay, so next are centrioles and microtubule. Okay, Okay, so micro, oh, this is a set. Okay, so central is this structure. Okay, so you can see that this is a microtubule. Okay, sorry, central is made of microtubule. Okay, so microtubule is just a single tube like this. Okay, so it is a structure that is only found in animal cells. Okay, typically they occur in pair. Okay, so they occur in pair. If you've learned mitosis before, you should have come across the term microtubule. Okay, so it's a pair of them. So it's a 9 times 3. It's a 9 times 3 organization. Okay, so we call this a triplet. So there are triplets of the microtubule, okay, which form a ring. Okay, and then that gives you a central structure. Okay, so it's a hollow cylinder, okay, 0.5 micrometer in length and 0.2 micrometer in diameter, so roughly. Okay, and also just FYI, and it is not membrane bound. Okay, so there are three, so if you are looking for a non-membrane bound organelle, central is one of them, the other one is ribosome. Okay, ribosome also does not contain any membrane, it's not membrane bound. Okay, so two of them lie at a right angle, okay, near the nucleus. They are mainly involved in cell division. Okay, so the microtubules within the centros form spindle fiber during nuclear division. Okay, just FYI, so spindle fiber, what is spindle fiber? Anyone? Okay, not the cell, but the chromosomes. Okay, so they extend from the central. So normally, during cell division, okay, so you have the central moving to both ends. Okay, and just FYI, this central is a very flexible structure. Okay, so if you think, let's say if this is a okay, let's say if this okay, your microtubule per se, if you zoom in, it's lots of protein. So let's say if you consider this as one protein, okay, so one bit is a protein, so you have lots of them. Okay, so of course I can't, so you have lots of them, then you form a hollow tube. Okay, so that's one microtubule, and this structure is very flexible, it can polymerize. Okay, so any one time you can add more bits, okay, then it lengthen, and also they can actually remove bit, then it shorten. Okay, so that's why your spindle fiber is essentially microtubules. Okay, so like I say, this is a very flexible structure that can polymerize, become longer, or it can also depolymerize, become short. Okay, so that's actually helps to move things around. Okay, microtubules generally are involved in movement. Okay, so yeah. So microtubules are straight unbranched hollow cylinder, 25 nanometer wide. Okay, very small. Okay, much smaller. So made of globular protein called tubulin. Okay, so one bit is one tubulin. So you have lots of tubulin. Okay, that polymerize to form a huge microtubule structure. So they make up what we call the cytoskeleton, which maintains cell shape. Okay, so here they use particular fluorescent protein. Okay, so the yellow one is a microtubule. Okay, so you can see that it's a cytoskeleton basically. Cyto is cell, okay, so they're the bone of the cell, so they help to maintain the structure of the entire cells. Okay, and also they're involved in movement of cell components within the cytoplasm. Later, I'm going to show you how is that the case, and it is also the components of spindle fiber during cell division. Okay. Next, cell surface membrane. Yeah.
Is that one relatively permeable?
Yes, selectively or selectively permeable. Yeah. Have you received a new schedule for next week? No. Okay, so for plasma membrane is probably something that we are familiar, one of the first cellular structure that you learned possibly. Okay, so so-called plasma membrane, so extremely thin, 7 nanometer thick. So again, it is a structure that you wouldn't see under a light microscope. Okay, so that very high magnification, okay, can be seen to have a 3-layer, 2 dark lines separated by a narrow line. Okay, so you need to have a very high magnification in order to see. Okay, so as you probably know, lipid bilayer with proteins embedded. Okay, so here this is one layer, so this is a phospholipid. I'm sure Okay, so your lipid bilayer is made up of the phospholipid. So the round shape is the phosphate group, okay? And then sometimes, okay, so whatever that's the two legs, okay, so that a fatty acid chain. So the entire thing is called phospholipid bilayer. You have many phospholipid arranged in one layer and then two layers. So that's why it is called phospholipid bilayer or simply lipid bilayer. And as you would note from this diagram, okay, so lots of all of these structures are proteins embedded within the layer. Okay, so we will have a chapter which will look at the plasma membrane in more detail. Okay, so some of the proteins embedded within the plasma membrane have been modified. Okay, so they've been modified to contain glycoprotein. Okay, so this serves as like an antenna for cell-to-cell recognition. Also, you have some cholesterol, okay, interjecting between your phospholipids. We'll learn in the future the function, okay, and yeah. So, and this membrane is selectively permeable. By selectively permeable, it means it allows certain things to pass through, but not the other things, okay? Anyone has any thought on what can, pass through this membrane directly? Water, okay, by osmosis, okay. Anything else?
Oxygen.
Yes, can, okay. And carbon dioxide, yes. Can hydrogen ion pass through this? Can hydrogen ion pass through? So basically anything, generally what is small can pass through, but can hydrogen ion pass through your plasma membrane? Do you know?
Yes.
It's a no, okay. So no, okay. And now make a guess. Generally, what is the what do you think distinguish this from this? Okay, so let's not talk about water first, but what do you think distinguish this oxygen, carbon dioxide, and hydrogen ion? Hydrogen ion is even smaller than this, okay, but how come hydrogen ion cannot pass through directly?
Well, I guess it's because there's charge.
It's charged, okay, and then why a charged particles cannot pass through a plasma membrane? Why? Any thought? Any thought? Okay. And anyone has any thoughts on what is a chemical nature of a phosphate group, okay, and fatty acid chain? How are they different in terms of?
One is what is the term for this?
So in biology, you probably learned the term hydrophobic and hydrophilic, okay? So this part is hydrophobic, this one is hydrophilic, okay? So hydrophobic refers to something that is non-polar, okay? So generally, whatever that is very charged, okay, so they are not welcome. So the hydrogen ion cannot pass through this layer, okay? So of course, we'll learn that in the future, okay? But I just wanted to know, so the selective permeability is partly attributed by this phospholipid bilayer, okay? So next, okay, function of cell surface membrane controls exchange of substances, okay, between the cell and its environment. So by substances, it can be waste product, can be oxygen, can be anything else, okay. So forms recognition site, okay, so that body's immune system can recognize its own cell, okay. So these all allow recognition. And for example, your red blood cells, okay, so they have this when we talk about blood group, okay. So blood group A, blood type A or B or O, okay. So it actually has to do with the glycoproteins which are present on plasma membrane, okay. So they act as receptor for attachment of hormones and neurotransmitter, okay. So and also they are folded to form microvilli, okay. So for example, you can see this is a membrane folding, okay. So your membrane folding, that is called microvilli. For example, in the gut cells, okay. So in the gut cells, they form microvilli again to increase surface area, okay. So you see that always membrane folding is for the purpose of increasing surface area and help cells attach to one another and form tissue, okay. So these glycoproteins, some of them also function to bind two things together, or bind two cells together. And then your cell wall and plasmodesmata. Okay.
So the phosphate group is it? Which one? Oh,
Yeah.
which one?
Phosphate group is for the round shape, okay? The round shape, okay? So
Like phosphoric, yeah, phosphoric.
PHOS, It is actually a sort of it's an anion of an acid. If you know phosphoric acid, H3PO4, you remove the H plus, that's the PO4. Phosphoric group is a PO4. Okay, so here's cell wall. Okay, so again, the structure you are very, very familiar with, only found in plant cells. Okay, so plant cell is made of cellulose. Okay, which is a very stiff structure. Okay, so they give the cell a definite shape. Okay, so that's why plant cells, because of the stiffness of the cellulose cell wall. Okay, so that's why plant cells normally have a definite shape. Okay, so it prevents bursting the cell from bursting. Okay, so if too much water enter, the cells can break. Okay, so the cell wall make sure that that won't happen. And cell wall per se is freely permeable. Okay, because even if you remember, so cell wall is basically it's like strand of a fiber, okay, wound together. So there's still space between them. So that's why things can just easily pass through. There's no membrane barrier. So that's why a cell wall per se allow free movement of molecules. Okay. And then between cell walls are not totally continuous. Okay. Sometimes there's some opening on the cell wall that's connecting two cells. Okay. But note that even though there is this opening, the cytoplasms are not continuous. Okay. So this space is still demarcated by membrane. Okay. So it is not the two cells, okay, the cytoplasms are not continuous. They are still separated. Okay. So they're still separated by plasma membrane. Okay, so but of course, with this channel, it facilitates transfer of materials between cells. Okay, for example, in the phloem cells, you learn that, okay, so you have materials being passed from one cell to the other. So that's a function of plasmodesmata. Again, plasmodesmata is a very, very fine structure. Okay, you don't normally see it on light microscope. Okay. And then permanent vacuole and tonoplast. Okay, so permanent vacuole. Okay, so vacuole is a large vesicles. Okay, normally they are more prominent in plant cells. Okay, so in plant cells normally you see a single large vacuole, but whereas in animal cells you see tiny ones. Sometimes we just call them vesicle rather than the term vacuole. Okay, so it's surrounded by membrane. So tonoplast is the membrane surrounding your vacuole. Okay, so it controls exchange between the vacuole and the cytoplasm. Okay, so again, this is a very flexible structure. For example, if water goes in, then it becomes like just like a balloon with water flowing in, it will just expand. So then it will push the cytoplasm against the cell wall. Okay, so it actually plays a function in regulating the turgidity of cells. Okay, sometimes, for example, you know that your plants, your leaves, for example, okay, maybe at night, okay, they are more flaccid, and then on daytime they are more, the leaves are actually more, what we say, the cells are turgid, so the leaves would actually stand up. Okay, so it's at least partly regulated by the vacuole. Okay, so then it is also a storage place. Okay, so all kind of water, ions, pigments, whatever that's okay, including your sugars which are synthesized from photosynthesis. Okay, so they regulate the osmotic properties of cells. So basically regulate the water content of a cell. Okay, so again, this is a and this would be your chloroplast, for example. You can possibly see some of the membranous structure. Okay, not very clear even with this. Okay, and Yeah. Oh, just FYI, did we mention about it? Just 1 minute. Let me see. Oh, and by the way, sometimes in your chloroplast, there can be starch granules. Yeah, starch granules can also be found in your chloroplast. Mhm. Okay, so are ribosomes visible under light microscope? No. Okay, why?
Too small.
Yeah, too small to interfere. Okay. So that's something you mentioned. Okay, so just this is a very commonly tested concept as to the distinction between resolution and magnification. Okay, so yesterday, as we say, resolution is about the ability to see two things which are separate as being apart. Okay. You know, recently, if you have read about, I heard from my friend, there is a viral post about a lady being able to catch her boyfriend cheating by looking at her phone records. So I think the story was that the boyfriend was, for example, on ground floor, and then she used a Samsung phone, and then she can actually zoom in and then can see the text on the phone. Okay, I don't know if you heard that. Okay, so but in any case, you can say that is a very, very high resolution phone, because if the phone is not high, the resolution is not high enough, even though if you zoom in, it's just it is just maybe a bright screen. Okay, you wouldn't be able to see the words, okay, as being separated. So that's an example of high resolution, because you can see that from afar, you can't really see the letters as being apart. Okay, so name the structure in plant cell and animal cells which can be seen with the electron microscope but not with the light microscope. Can you name a few?
Ribosome.
Yeah. Yes, like ribosomes. Yes. Okay. Anything else? Ribosomes. Yeah, you can write like ribosomes.
Membrane.
Okay. Okay. Plasma membrane. Actually, yeah, you can't really see the plasma membrane with light microscope. Anything else? Ribosomes. She's mentioned that.
Or say lysosome.
Oh, lysosome. Okay. Yes. Yeah, lysosome potentially. Yes. Okay. So anything that's membrane bound. Okay. Actually, yeah, anything that's membrane bound and also non-membrane bound. Okay. So your Ravier, smooth, all of this structure can be seen under the electron microscope. Yeah, chromatin, as we learned yesterday, chromatin is just basically chromatin chromosomes are the same thing. It's just a different level or different degree of organization. Okay, so one is highly wound, okay, the other one is more loosely bound. Okay. Okay. So okay, so it's never I've already shown you the answer. Okay, so centrals are only found in animal cells, but not in plant cells. Okay. maybe you can answer the next 3 questions. I'll go into it.
Okay, go ahead. Three organized boundary and because in the boundary is just danger. 这个打过去被骂的很凶，买过去就买过去就boundary 那是说我在这上面上面，它是平时很被它往里面。外国风的嘛，我是从国际出来的，它这里很安稳，被吧。外国风是那，外国风是来的，我都没有国际。对，我来国风是那个什么，那个花钱的啊，来跟他们，你们写的打工码都不学是吧？那个就是这个当感觉那个从那个纸身上的，哦，还有这么，
Okay. So your those lacking boundary membrane. Okay, so here are the structure. Ribosome centrals, as you mentioned. Nucleolus as well. Okay. Yes, nucleolus. Okay, so single membrane bound organelles. Okay, so they are here. And double membrane only mitochondria, chloroplast and nucleus. Okay, so
is nuclear envelope count?
is nuclear envelope what?
Count.
Yes, it's actually yeah. So nucleus is bound by a double layer membrane. Yes. Okay. So next we'll learn about ATP. Okay. So anyone remember what's ATP? Sorry? Yeah. Okay, adenosine triphosphate, what does it do? Yeah. So it is the energy source itself. Okay. So it is actually the energy source. Yeah, in a way you can say that it supplies energy, okay, but it's a storage form of our energy. Okay. So ATP, so it is produced, okay, so we know it's generally in plant cells, it can be produced in chloroplast mainly during cell photosynthesis, okay. So and of course in mitochondria, where in animal, actually in plant cells as well, okay. So it's found in both animal cells and plant cells.
If you have stepper.
Yeah, I don't actually have a stepper. Yeah, so okay, so okay, so ATP is called a universal energy currency. Okay, so no matter it is eukaryotes or prokaryotes or whatever cells, okay, so their energy ATP is used as a general energy storage form. Okay, so you can use energy to ATP to provide energy. Yeah, very often we say some compounds store energy, for example, like fuel, okay, for example, you know, the petrol that we burn every day, okay, so because when we say a compound store energy is when you break the bonds inside, okay, so lots of energy is being released, okay. So in the case of ATP, actually, so this is a structure, general structure of your ATP. So T stands for triphosphate, okay? So of course, if you remove one, then it becomes a diphosphate. Remove another one, then it becomes monophosphate, okay? So whenever this bond is broken, okay, energy will be released, okay? So it is the amount of energy that's released that's used to do work, okay? So it's ATP itself is not a form of energy, okay? But when you break it, it releases the energy, okay? So yeah, that's okay. So and one key characteristic of it is that it is very small and water soluble. So because it is small and water soluble, it can easily spread around cells. Of course, this is by diffusion. Okay, your cytoplasm is largely a watery environment. Okay, so anything that's water soluble will move around a watery environment like the cytoplasm very quickly. Okay, so yeah. So when we say it is a universal, okay, it means in almost all cell types. Okay, so for example, both your eukaryotes and prokaryotes. Okay, so eukaryotes and prokaryotes are one of the very general categorization of cells. Okay, so but basically they should encompass all cell types. Of course, we don't consider virus as a cell. Okay, but yeah, with virus as an exception. Okay, but the rest, so they're all eukaryotes or prokaryotes. Okay, so all of them use ATP as an energy source. Okay, you can see that it is something that is what we say evolutionary conserved, which means everything that evolves from that single species still uses this. Okay, so it is a highly conserved structure. Okay, so next prokaryote versus eukaryotes. Okay, so we kind of have some discussion about this. Okay, so prokaryotes are generally the more primitive organisms. Okay, primitive mean how should I say, older in origin, like yuans. Okay, okay, so something that's more primitive. Okay, okay, so as you mentioned, okay, the term you, okay, by the way, you means true. Okay, yeah, so anyway, so basically two nucleus. Okay, so organisms whose cells possess nuclei. Okay, so they are the eukaryotes. So prokaryotes, they lack nuclei. Okay, so and eukaryotes contain many membranous organelles. Okay, so as we have learned, your Golgi, mitochondria, chloroplasts, lysosomes, all of these are membranous structure. Okay, but then actually okay, at least for your prokaryotes, they do not have at all any double membrane bound organelles, but they may form some vesicles like lysosomes. Okay, and eukaryotes can be both unicellular or multicellular. Okay, so yeast is unicellular, for example, or if you know the Plasmodium, which causes malaria, is unicellular. Multicellular, there are plenty of that around us. Okay, and ourselves. Okay, and then and of course your prokaryotes are much smaller than your eukaryotes. Okay, so have a single, so for eukaryotes, okay, normally your DNA is highly packaged into chromosomes, for example, but whereas in prokaryotes, normally it's just a circular chromosomes, okay, and or circular chromosomes or DNA just present in cytoplasm, not membrane bound, not tightly packed, okay. So okay, so of course we believe that eukaryotes are evolved from prokaryotes, okay, and so example prokaryotes are bacteria. Hold on. Maybe just add a note, okay, your genetic material. Okay, so everything, genetic materials are packaged inside your nucleus, okay? So whereas, okay, your in the prokaryotes, it's just loosely floating in the cytoplasm, okay? So it doesn't, it's not membrane bound, okay? Okay, so here as we see, your prokaryotic cells are mostly unicellular. Okay, so the diameter is very small. Okay, 0.5 to 5 micrometer. Sometimes, okay, so sometimes they always have this cell wall. Okay, so the cell wall are made of peptidoglycan. So if you look at this term, it is made of amino acids and sugar. Okay, so the peptido that's come from amino acid, glycan come from sugar. Okay, so the cell is made of peptidoglycan. Okay, they lack organelles. Okay, so maybe like I said, sometimes you may see some vesicles. Okay, and then the few organelles. Okay, and also lack double membrane bound organelles. Okay, have naked circular DNA, including plasmid. Okay, so actually, these and these, they are both circular DNA. So when we say circular, it means, you know, the double strand. Okay. So your double strand of DNA, okay, so they just form into a circle. Okay, so let's say this looks like a double strand. Okay, anyway, so this is just much larger in size, as in like the entire length. Okay, so plasmid is a smaller circular DNA. Your general chromosomes is a larger circular DNA. In any case, they're circular in nature, as in they are formed into a loop. Okay, but in eukaryotic cells, it is not. So the both ends are open. They're open-ended in eukaryotic cells. Okay, and it contains small 70S ribosomes, as mentioned before. So you can see that some of these structure are always present in bacterial cell or in prokaryotes. Okay, but they may have additional structure, such as first flagellum. Okay, so flagellum is like a tail. It's mainly for movement. Okay, for locomotion. Capsules are some additional material that are secreted on the outside. They serve as a protection. So generally, bacteria which has capsule tends to be more infectious, tends to be harmful. Okay, so yeah, and partly because this capsule provide protection, therefore they won't die, and then they cause problem to us. Okay, so sometimes they may have infolding of plasma membrane. Okay, so for all kind of purpose. Okay, sometimes for example here, for photosynthetic membrane or carry out nitrogen fixation. Okay, so then plasmid. Okay, and sometimes present, sometimes not. Pili are this also this structure. Okay, of the pillars is the singular form. Okay, so involved in sexual reproduction. Basically, just allow two cells to come together and stick together. Okay, so this endosymbiont theory. Okay, so it's something that I have mentioned. Okay, and okay, so as we learned before, your mitochondria and chloroplasts, they are unique in eukaryotic cells in the sense that they contain 70S ribosomes and circular DNA, which are not normally present in eukaryotes. As I mentioned before, okay, so they were initially independent living prokaryotes, which at some point, you know, they just come to live symbiotically with the eukaryotic cells. By symbiosis, you probably learned that in high school, okay? When two organisms live together and then they confer a mutual benefit to each other, then that's kind of relationship is called symbiosis. Okay, so and the DNA contained within the within the mitochondria or the chloroplast synthesize some proteins which are only uniquely found on mitochondria and chloroplast. And finally, viruses. Okay, so they are very small. Sometimes are not necessarily categorized as cells. Okay, so do not have cell structure, and sometimes yeah, they're just a disease-causing agent. We do not consider them as being an organism. Okay, so the only characteristic that it has that is common to other living things is that they can reproduce, but they do not respire. They do not, you know, they do not undergo excretion. Okay, yeah. They are even smaller than bacteria, so normally in nanometer range, the size. Okay, and commonly first they have this protein. Okay, so a protein structure that surround the genetic material. Okay, so there is so the genetic material can be DNA, can be RNA, And the DNA can be single strand or double stranded. Okay, so and then on the protein membrane, so we have this structure called the spike proteins. If you have heard about, during COVID, we always talk about so mutations or variations in the COVID. Okay, very often it's because of the change in the structure of spike proteins, which makes your antibodies no longer, or your original vaccine no longer useful. Okay, can you please tell me why the mutation would render it no longer would actually make the original protection afforded by a vaccination useless? Why? Why a mutation would make it, not responsive?
Percent is because they recognize the previous protein structure.
Okay, but when we say recognize, what does it mean? When we talk about human, I can recognize your face. Okay, but how does recognition happen at the molecular level? What do we mean by shape? Okay, so basically, again, something we'll keep coming across in biology is that all of the recognition, all of the interaction is very shape dependent. Okay, so of course, like enzymes or active site. Okay, so anyway, so for example, originally this is a spike protein. Okay, and then so if this is a spike protein and then this is the antibody, so if the spike protein suddenly change shape, then the antibody can no longer bind to it, and that's why mutation can actually mutation of the surface membrane protein, okay, or the surface protein like spike proteins can actually allow an organism and a pathogen from allowing to escape from our immune system. Okay, so and also it has a protective coat or protein molecule called the capsid. Okay, so this is coat rather than coat. Okay, so yeah, that's it, and I think we'll continue later. Okay, so I'll see you later for at 10. Okay. Okay. Okay, so this is what we call the eyepiece. Okay, so of course, in the eyepiece, if you look it down, okay, under the view, you may see something like this. Okay, sometimes depending on how it is set up. Okay, so under the eyepiece, you can see this what we call reticule. Okay, so of course, having it is actually is very convenient for us to just by looking down under the microscope, we know immediately roughly what is the size of a particular structure. Okay, so normally we use this eyepiece reticule for that purpose. So it has a transparent scale. It usually has 100 division. Okay, but the thing is that we said it is not so hold on. We will know. So what is the calibration? As in, so here we know it is 100 divisions on the eyepiece reticule, but each division, what is the size of each division? For example, we do not normally know. Okay, so that's why we rely on another piece of instrument called the stage micrometer. So normally under the microscope, okay, you can actually you can also put a little scale. Okay, so basically you just, so let's say if this is a slide. Okay, so normally on the scale stage micrometer, so it is another scale, so which is this. Okay, so this is on under the objective lens. Okay, so this is like a sample, and then on the eyepiece, when you look down on it, so the reticule on the eyepiece will be juxtaposed, basically placed next to your stage micrometer. Okay, so for stage micrometer, we know what is the division. Okay, for a stage micrometer, typically this is like 0.1 millimeter. Okay, one large division, and every small division is therefore 0.01 millimeter. Okay, so of course it'll be 100 micrometer and a 10 micrometer. Okay, so what you do, okay, so for example, in this particular case, for this eyepiece reticule that is used, because sometimes different eyepieces come with different reticule. Okay, so you need to do your calibration. Okay, so here under this case, okay, so we see that so here, yeah, 2, 3, 4. There are 40 divisions on an eyepiece reticule, and that correspond to 0.1 millimeter. Oh, hold on. Am I right? So, so this is 100 unit. Okay, so you can also see. So, of course, sometimes for this one, it is well aligned, but you can actually just look at this part as well. Okay, so for this one, we know that 100 unit here correspond to 0.25 millimeter. Okay, so you can just do the maths. Okay, so the eyepiece unit, okay, so this is a measurement of one eyepiece unit. Okay, so that is like 0.25 millimeter. So you have to say this 0. Can you please tell me? Sorry, actually, this is the wrong something. This should be divided by 100. Okay, so this should be divided by 100. Out. So of course, depend on what you. Just 1 minute. I'll check on that. Okay, so if you look at this case, so here there are 100 divisions. Okay, so here there's 100 division, which is why you divide that by 100. Okay, so because there are 100 divisions on your eyepiece unit. Okay, on your eyepiece reticule, that is the reason why we divided by 100. Okay, so this whole thing is not 0.25 micrometer. Okay, so then you will calculate that each unit. Okay, so if you zoom into this, okay, so each unit here is 2.5 micrometer. Okay, if you do the conversion, so each of them is 2.5 micrometer. And then here, if you look at this, so this one is 20 eyepiece reticule unit. Okay, so we know 20 units is, so each unit is 2.5 micrometer. So we can just do the maths. Okay, so it's actually a very simple mathematical calculation. Okay, are we good there? Okay. So next, maybe, okay, so just FYI before I continue, okay. When you do what we call calibration, you can also maybe just pick a certain region. For example, you can also do here. Okay, so for example, here we see for 0.1 millimeter, that would correspond to how many eyepiece reticule unit? That would be from here to here. Okay, that is 40 unit. Okay, you will get the same number. Okay, you will get the same number, which is also 0.005. Okay, so it's up to you where. So as long as you pick somewhere where you can easily determine, okay, whatever unit on a stage micrometer correspond to whatever number of units on the eyepiece reticule, that is fine. Okay, so here, okay, can you please do this? Can you please do the next one? Okay, you can probably do the calibration for this one. Okay. Using similar method just now. Yeah, I'll give you 3 minutes to try the two MCQ, the question down there, okay? So, question one would relate to this as well, okay? So, question one would relate to this diagram. Question two is a separate, different, IP strategy. Okay, so let's have a look here. So for this one, okay, so for one eyepiece reticle unit, okay, so let's have a look. How many units? Okay, so here this is 0.1 millimeter. Okay, so that correspond to how many eyepiece reticle unit? 100. Okay, so it's because okay, it happens to span the entire length. You just divide it by 100. Of course, if you want to convert that into a micrometer scale, so from millimeter to micrometer, you times 1,000. Okay, you divide by 100. Okay, so cos one eyepiece unit, okay, equals to 10 micrometer here. Okay, so if you look at what we did here, okay, so the answer would be C. Okay, because what you did was multiply that by 1,000, divide by 100. Okay, so that was how we obtained the unit, the length of one eyepiece reticle unit. Okay. And next for this one, you will go with the similar. Okay, so here what we see is that, okay, so here for, okay, let's say it's up to you. So let's say if you look at here and here, okay, so what is the length? Okay.
Division.
For the stage micro, okay, of course, this would be the stage micrometer. This is a stage. What is the length there?
0.2 mm.
Okay, so that will be 0.2 millimeter. Okay, and how many units are there that correspond to? So 0.2 millimeter correspond to how many? 80 units. Okay.
It's 8.
So okay, by unit we means this small division.
Oh,
Okay, so that will be, so just like you divide by 100 just now, because there are 100 small divisions. Okay, so that will be 80 division. Okay, so of course if you want, you can convert that into 200 micrometer divided by 80, so that will be 2.5 micrometer per unit. Okay, which if you are looking at the area, so which means you need to know. Okay, so for area, you need to know the radius here. So your radius will be equal to what? Your radius will be equal to, okay, how many units? How many eyepiece vertical unit for your radius? No, radius.
Oh,
This is diameter, right?
yeah.
So radius will be 50 eyepiece vertical. Okay, so 50 vertical unit, which also equals to 50 times 2.5 equals to 125 micrometer. So that is the radius. Okay, so here we do step by step. First, we need to know what is the size of each unit. Okay, so from there, you do the calculation. We know that it's 2.5 micrometer. So here the radius is 50 vertical unit. So you note that the radius is actually 125 micrometers. So of course, with the area pi r square, your answer will be C. Okay, so that will be it for this particular chapter. Okay, so we're doing a bit of a tutorial question, but before that, actually, I realized there's something which I haven't briefed you on. So okay, just so this is something I still need to finalize and send it to you, but roughly. Oh, before that, actually, okay, so I want to show you a video. Okay, before we continue, okay? Okay, so here's the structure of a cell. Okay, so I just wanted to see this. Hold on. So these are whatever glycoproteins in the plasma membrane that allow cells and cells to interact with each other. So you can see these two different cells. Okay, this is an interface of two cells, glycoproteins connected with each other. Okay, so this is a plasma membrane. The bed there is a plasma membrane. So the phospholipid bilayer, you can see that it is a very dynamic structure. Things can move around. And this part, okay, you see my cursor? Yeah, you can see my, so this is like, proteins or whatever, okay, that can move around the plasma membrane. So there's some part which is protruded to the outside. So that's viewing from the inside, okay? So actually from inside the cells, you can see that there are some structures of those strands. Okay, so they're also cytoskeleton. They help to keep the shape of the cell. Okay, you just zoom down into going within the cells. Okay, so these are the cytoskeleton. So just some structure protein which keeps the shape. Okay. So this is a protein synthesis. Things are being assembled. Actually, that's active. What do you guess that is? That yellow thing, this thing, what is this? What do you think it did? So this is a bit like an enzyme, basically just, just chop things off like Caesar, cut things, okay? And what do you think this is? A hollow tube, which can lengthen. What is that? Microtubule, okay? So this thing. Okay, so you can see that it can actually, that's how it polymerizes, and it can also depolymerize where it becomes shunted. Okay, so as we mentioned just now, your microtubule is also involved in the transport of stuff. Okay, so actually what you see here, this big bubble, that's the vesicle. Okay, so like inside the cell, okay, so because you have to remember that inside the cell, everything is all they have no break. It's not like us, we can walk, we can see, or I want to walk to a set direction. Okay, so how can things be directed within the cell? Okay, so it's very much partly attributed to the presence of this microtubule. Okay, so then you have the vesicles, you have these proteins that you see, it looks like it has a leg. Okay, so let's the structure is beyond what's its name is beyond your scope, but basically you can see that there are proteins which will drag the vesicles along the microtubule. So actually you see, what is that? What is this? What do you think this is? Sorry? What is that? So there are two tubes, two big hollow tubes. What are they? Centrioles. Okay, so those are the centrioles. This is likely to be a mitochondria. Okay, you have lots of these extending. So these are nuclear pore. Okay, I think yesterday I mentioned your nucleus is like the CPU. Information is taken out. So your mRNA, the pen drive, that's it. So we say that you need to have nuclear pore to allow mRNA to leave the nucleus. And then once they leave the nucleus, they will be bound to the, okay, let's see, ribosomes. Okay, the green one is the ribosomes. Okay, if you see two things come together, so we mentioned that ribosome has two subunits, a smaller and a large one. Okay, combined together, it forms a protein machine synthesis machineries. Okay, which would then, okay, it will move along the mRNA to translate the protein. So this yellow bit is a protein. Okay. That's a mitochondria. Okay, so of course, sometimes, so this is like, for example, this is protein synthesis. This is going into, for example, the rough ER. Okay, so the proteins synthesize, okay, these are ribosomes and the rough ER. When the protein is translated, it goes into the ER membrane space. So what happened is that this protein will pinch off as vesicle from the ER. So this is, what structure is this? Golgi. Okay, so you can see that the vesicles from the bottom is coming from the ER. It is going to fuse with the Golgi. Okay, and then things are coming off, budding off from the Golgi. Okay, so these vesicles are budding from it. Okay, so ultimately it will be transported to a plasma membrane. Okay, and then so we mentioned that, so the content will be released to the outside, and also I mentioned that some of the proteins are embedded on the membrane. Okay, so whatever proteins to be exported outside or to be embedded within the membrane, very often they come from the rough ER. Okay, so these are just recognition. They may come together, these proteins may come together. So they form connections, so cells may be bound to each other. This is not cellular structure, but not the internal structure, but these are just some pipe that cells we can change shape, squeeze through your blood vessels into tissue. Okay, so that's what you see there. Just blood vessels. Okay, so you can see lots of that. Okay, so that's it for that. Okay, and just one last thing that I should have mentioned on our first day is, what's the subject? Okay, hold on. I will send you a finalized version. Where is that? Okay. So roughly for what will happen for biology, of course, we have yeah, perhaps. So actually, this is for the Gen intake, but I think I'm getting ahead of the schedule. I'll be finishing the 11th topic for AS by end of June, actually, by May, actually. Okay, so any case, so you have, like I mentioned the other day, you have 11 AS topic and then 8 A2 topic, which will be finished over the year. Okay, so I think for at the end of, I think for your case, at the end of October, so for your case, you have at the end of year, you have an exam. And before that, there will be two tests by the end of this year. Okay, so of course, it depends on whether you want to take your AS paper or not. Okay, so actually in September, we have one round. So actually, sorry, by September, there'll be one round of tests. Okay, so of course, if you want, it is possible for you to take AS paper, but I think might be too rushed. Okay, but for mathematics, definitely you can. Okay, so if you want, you can do your maths or AS in October, perhaps for physics. Okay, but generally, for each semester, there'll be two tests. Okay, so amounting to like 50% of the test may not, the full mark may not be 40. Okay, so that's to be changed. Okay, and then there'll be a final exam, but of course in a slightly different timeline for you guys. Okay, and then so there'll be so there'll actually be 3 papers for the exam. I think they will likely happen at the end of the year. Okay, so there'll be 3 papers, paper 1, paper 2, and then paper 3 as a practical. Okay, and then semester next year you'll be doing another 2 tests and then 1 exam. So in terms of the calculation, actually I still need to finalize it, but at least I want you to expect that by the end of the year there'll be 2 small tests, 1 major exam. Okay, so and then next year also 2 tests and then 1 major exam. To be honest, whatever results that you score for your tests within the institute doesn't matter as much, in the sense that, okay, your final A-level results will really be just based on your actual performance at the exam. And generally, of course, you'll be provided forecasts for your exam results here. And at the institute, the grading is more strict. Okay, for example, perhaps for example, if you want to go score an A, you need to score above 70 mark for certain paper. But if you look at the grade threshold for actual Cambridge exam, for some papers, 16 marks, you can score A already. So because some papers are more difficult, more challenging than the other. Okay, so of course they adjust it every year. But I think on average, for example, paper 4, which is the toughest one, okay, the average mark, the raw mark to score A is around 60-ish. Yeah, and then for paper 1, the raw mark to score A is not 32 more or less. Actually slightly above 30. So yeah, but in any case, for internal exam, you have a stricter grading threshold. Okay, so that will be it for this